Hello, everyone. Today, we will introduce cancer immunotherapy. There are 4 parts: introduction of cancer immunotherapy, types of cancer immunotherapy, F.D.A. Approved antibodies, immunotherapy by cancer type. Cancer immunotherapy, sometimes called immuno-oncology, is the artificial stimulation of the immune system to treat cancer, improving on the system's natural ability to fight cancer. Immunotherapy agents do not directly attack the tumor but instead mobilize the immune system. This can be achieved through various approaches that utilize adaptive or innate immunity. The main types of immunotherapy can be broadly subdivided into non-antigen-specific and antigen-specific categories. Non-antigen-specific strategies include non-specific immune stimulation and immune checkpoint inhibitors, whereas antigen-specific strategies include adoptive cell transfer of autologous cancer-specific T-cells, and various therapeutic vaccination approaches. Non-specific immune stimulation can be achieved with agents that stimulate immune effector cells such as T-cells and APCs, for example, DCs, or inhibit and/or deplete immunoregulatory cells, such as Treg cells. Immune checkpoints are proteins in the immune system that either turn up a signal or turn down a signal. These proteins help keep immune responses in check and can keep T-cells from killing cancer cells. When these proteins are blocked, the brakes on the immune system are released and T-cells are able to kill cancer cells better. Adoptive T-cell therapy is a form of passive immunization by the transfusion of T-cells, adoptive cell transfer. They are found in blood and tissue and usually activate when they find foreign pathogens. Specifically, they activate when the T-cell surface receptors encounter cells that display parts of foreign proteins on their surface antigens. These can be either infected cells, or antigen-presenting cells, APCs. Such as figure, cancer-specific T-cells can be obtained by fragmentation and isolation of tumor-infiltrating lymphocytes, or by genetically engineering cells from peripheral blood. The cells are activated and grown prior to transfusion into the recipient, tumor bearer. Based on their use and act, we will focus here on the tumor immunology of T-cells specifically. To mount an effective and targeted response, T-cells must be able to recognize and target specific antigens presented in the context of major histocompatibility complex, MHC, proteins on the tumor that are not present or are poorly expressed on healthy tissue. Tumor-specific T-cells can recognize overexpressed antigens, neoantigens derived from germline mutations, or so-called cancer germline antigens expressed de novo during carcinogenesis. However, several processes exist to suppress anti-cancer responses. T-cell intrinsic mechanisms such as loss of functionality and expression of checkpoint proteins, PD-1, CTLA-4, lead to T-cell exhaustion. Tumor intrinsic mechanisms include secretion of suppressive factors such as TGF-B, or expression, or checkpoint ligands. Furthermore, tumors recruit suppressive cells such as regulatory T-cells and tumor-associated macrophages that further inhibit T-cell responses. How therapeutic cancer vaccines work. Therapeutic cancer vaccines can be designed in a number of ways. Tumor antigens can be directly loaded onto the immune system's antigen-presenting cells, dendritic cells, outside the body and then reintroduced. Or they can be administered in combination with viruses, bacteria or other substances that stimulate the immune system, adjuvants, to get dendritic cells to pick them up in vivo. Either way, the dendritic cells then travel to the lymph nodes to prime and activate T-cells, so they can hunt down cancer cells expressing the tumor antigens. There are many antibodies were approved by FDA for treatment of cancer, such as alemtuzumab, atezolizumab, avelumab, ipilimumab, ofatumumab, nivolumab, pembrolizumab, etc. From the preventive vaccine for cervical cancer to the first therapy ever proven to extend the lives of patients with metastatic melanoma, immunology has already led to major treatment breakthroughs for a number of cancers, such as bladder cancer, brain cancer, breast cancer, cervical cancer, childhood cancer, colorectal cancer, esophageal cancer. Every cancer type is unique, though, and immunology and immunotherapy are impacting each cancer in different ways. BOC Sciences can provide wide range of target inhibitors and agonists to help your research, such as EDO, PDL1, CTLA4, CD39 and so on. BOC Sciences is specialized in the inhibitor products and services, providing a confidential and efficient products at competitive prices. Every step of peptide synthesis is subject to BOC Sciences' stringent quality control. The exceptional quality and reliability of our peptide modifications service has been appreciated by customers worldwide for many years.